We are going to talk about the river of life inside you right now.
It really is the river of life. And I know that sounds like a cliche, but when you actually dig into the physiology of blood, you realize that river is almost too passive a word. It's a highway, it's a waste management system, it's a communication network, and it is a battlefield.
Absolutely. And specifically, we aren't just talking about blood in the general, liquid sense today. We are zooming in on the single most common cell in that fluid,
The red blood cell.
the red blood cell or the erythrocyte.
That's right. And this is really a masterclass in human physiology. If you understand the red blood cell, how it's born, how it works, and how it dies, you basically understand the fundamental constraints of human life.
That's a big claim.
It is, but I think it holds up. We're going to look at the life cycle of these cells, the absolutely insane chemistry required to build them, and the devastating medical conditions that arise when even one tiny part of this system breaks down.
Right. I have the source material right here, chapter 33. And honestly, the first thing that jumped out at me wasn't the biology. It was the engineering.
The engineering, how so?
The text makes a pretty bold claim right out of the gate. It calls the red blood cell a bag.
Yeah.
Which, I don't know, it feels a little derogatory for something that keeps us alive.
It does sound unscientific, doesn't it? A bag. But it's actually the most accurate engineering description we have. You have to strip away the idea of a standard cell. You know, the picture you drew in high school biology with a nucleus in the middle, mitochondria, ribosomes, all that furniture.
Right, all the organelles.
All the organelles. The mature RBC has stripped all of that out. It is literally a flexible membrane holding a fluid matrix. It's a purpose-built container.
And that flexibility, that bagginess is the whole point.
It's everything.
Because I was looking at the dimensions here. The mean diameter of a red blood cell is about 7.8 micrometers.
Right. Keep that number in mind, 7.8.
But the capillaries, the tiny roads this truck needs to drive down, can be as small as 3 or 4 micrometers.
Exactly. So do the math. You have a 7.8 micrometer object that has to fit through a 3 micrometer hole.
It's impossible.
It would be. Imagine trying to drive a semi-truck through a tunnel that is half the width of the truck. If the cell were a rigid sphere, like a marble, it would clog the system instantly.
You'd have a massive block.
A traffic jam at the cellular level, you'd get tissue death, potential stroke, all within seconds. The whole system would fail.
So it needs to squash. It needs to deform.
It needs to do more than squash. It needs to deform completely without stretching the material to the breaking point. This is where the text highlights a concept called excess membrane.
Excess membrane, which implies it's wearing a coat that's like 2 sizes too big for it.
Precisely. The cell isn't filled to capacity. If you have a sphere, the surface area is minimized for that volume. You can't change its shape without stretching the skin, right?
Right, like a fully inflated balloon.
But the RBC is a biconcave disc. Imagine a donut without the hole punched all the way through, just pinched in the middle. That shape provides a massive amount of extra surface area relative to the volume inside.
Okay, so it's like a deflated beach ball. You can grab one side and squeeze it through a chain link fence because the skin is loose.
That is the perfect analogy, and that's exactly what it does. It folds over on itself to get through those tight squeezes.
And that shape, the biconcave disc, it must have other advantages too.
It does. It serves a dual purpose. Yes, it allows for that extreme deformation to pass through capillaries, but it also maximizes the surface area for gas exchange.
Ah, of course.
Oxygen doesn't have to travel far to get to the center of the cell because the cell is thin in the middle. It diffuses in and out much more rapidly than it would from a sphere of the same volume. It's optimized for speed.
Okay, so a brilliant piece of biological engineering. Now we need to talk about what's inside the bag. The obvious answer is hemoglobin.
Right. The primary definition of an erythrocyte, if you had to give one, is a vehicle for hemoglobin. That's its whole reason for being.
But here is a nuance from the text that I found surprising. In lower animals, some invertebrates are mentioned, hemoglobin doesn't need a cell. It just floats around freely in their version of blood.
This does.
So why do we need to package it? Why the extra step? Why build this whole complicated bag for it?
It comes down to 2 big problems: efficiency and pressure. Let's take efficiency first. If hemoglobin were free in human plasma, the text says about 3% of it would leak through the capillary membranes into the tissue spaces every single time the blood passed through.
3% per cycle. That's a huge loss.
It's an enormous loss. It's a small molecule relative to the pores in our capillaries. So we'd just be constantly leaking this vital protein into our tissues, or worse.
The kidneys would filter it out?
Exactly. It would end up in the urine. We couldn't hold on to it. But there's a physics reason, too, which is maybe even more important.
Osmotic pressure.
Osmotic pressure. If you dissolved that much protein directly into the plasma, the colloid osmotic pressure of your blood would be unmanageable. It would suck water out of your tissues constantly. Your fluid balance would be a catastrophe.
So by packing it inside a cell, you effectively hide it from the fluid dynamics of the plasma.
You're containing its osmotic effect. The cell membrane handles that, not the plasma itself. It's a way to cheat the physics of solutions.
That's incredible. Okay, so oxygen transport is job one.
Hmm.
But that's not the whole story, is it? Did you catch the hidden function in the source?
Oh, yeah. The bonus function?
Carbonic anhydrase.
Yes.
It sounds like a chemical weapon, but apparently it's an absolute hero.
A massive hero. Everyone focuses on oxygen delivery because that's what we associate with breathing. But waste removal is just as critical, maybe more so, minute to minute.
We produce CO2 constantly.
Massive amounts of CO2 in our tissues from metabolism. And CO2 doesn't dissolve into water or blood plasma very well on its own. It's a very slow, inefficient reaction.
So if we just relied on the plasma to carry CO2 to the lungs, we'd be toxic in minutes.
We'd be dead, absolutely. The pH of our blood would plummet. So inside the red blood cell, sitting right alongside all that hemoglobin, there is a massive quantity of this enzyme, carbonic anhydrase.
And its job is...
What, exactly?
Its job is to catalyze the reaction between carbon dioxide and water to form carbonic acid.
And when you say catalyze,
Yeah.
you mean speed it up.
I mean, it accelerates the reaction rate about 5,000-fold. It makes it nearly instantaneous. As soon as CO2 enters the red blood cell, bam, it's converted.
Into what?
Into carbonic acid, which then immediately dissociates into a bicarbonate ion and a hydrogen ion. And that bicarbonate ion is super soluble, so it can be transported easily in the blood.
So the RBC is like a chemical processing plant. It picks up the trash, compacts it chemically so it fits on the truck, and then transports it to the lungs. That's a great way to put it. It allows the water in our blood to transport enormous quantities of CO2 that it otherwise couldn't handle.
And then in the lungs, it just reverses the process.
Instantly. The low CO2 environment in the lungs pushes the reaction the other way. The enzyme helps it go back to CO2, gas, and water so you can exhale it. Without that enzyme, you suffocate on your own waste.
And there's also a note here about it being an acid-base buffer. That's the third job.
That's the third function, and it's tied directly to the hemoglobin itself. Hemoglobin is an excellent acid-base buffer. All those hydrogen ions produced from the carbonic acid reaction, hemoglobin soaks them up.
So it prevents the blood from becoming too acidic.
Precisely. In fact, the red blood cells are responsible for most of the buffering power of whole blood. They are the primary reason your blood pH stays so incredibly stable, right around 7.4.
Okay, so let me get this straight. It's a delivery truck for oxygen, a hazmat team for CO2, and a chemical stabilizer for pH.
Multitasking at the cellular level, it's an incredibly efficient little package.
Let's get a sense of scale here, because microscopic is a word we use. But the numbers in the source material are... they're hard to wrap your head around.
They are astronomical. It really is a numbers game. Let's look at the density, the concentration of these cells.
Okay.
In a healthy man, in just 1 cubic millimeter of blood, which is a tiny drop the size of a pinhead, there are about 5,200,000 red blood cells. Give or take a few hundred thousand.
5.2 million. In a single drop of blood.
Correct. For women, it's slightly lower, about 4,700,000. But that density is staggering. Think about the total number in your body. It's in the trillions.
And this brings us to a term you see on doctors' reports all the time, hematocrit.
Right.
I usually just gloss over that column. I see the number, I see the normal range, and I move on.
A lot of people do. But it's a really simple, powerful physical measurement. Hematocrit is just the percentage of the blood that is actually cells, the solid part.
How do they measure that?
It's very low tech, actually. You take a test tube of blood, a little capillary tube, and you spin it in a centrifuge. The heavy cells, the red blood cells, pack down to the bottom. The lighter plasma stays on top.
And you just measure the ratio.
You measure the ratio. In a normal person, that packed red cell volume is about 40 to 45% of the total volume.
So my blood is roughly half liquid plasma, half solid cells.
Give or take. And that number is hugely important. If it drops, you're anemic. If it goes way up, you have polycythemia, which we'll talk about. It's a fundamental measure of your blood's oxygen-carrying capacity.
And inside those cells, there's another limit, right? The amount of hemoglobin you can pack in.
Right. It's not infinite. You can't just keep stuffing more in.
It's like trying to stuff a suitcase.
Exactly. There is a hard metabolic limit. The cell's machinery can concentrate hemoglobin up to about 34 grams per 100 milliliters of cells. It physically cannot go higher than that. It's just the metabolic limit of the cell's hemoglobin-forming mechanism. The molecules are a certain size, they need a certain amount of water around them. 34% is the maximum packing density you can achieve.
So that's the theoretical max. And in healthy people, we're pretty close to that.
We are. In healthy people, the concentration is almost always at that maximum. But in certain types of anemia, the cells might be produced with only half that amount of hemoglobin. They're the right size, but they're half empty.
They're empty bags.
They're functionally empty bags. And this all translates directly into how much oxygen your blood can carry. The text does the math.
Right. Each gram of pure hemoglobin combined with 1.34 milliliters of oxygen.
So you multiply that by the total grams of hemoglobin in your blood, and you find that a healthy man can carry about 20 milliliters of oxygen per 100 milliliters of blood. For women, it's about 19.
And that's the number that matters. That's the actual payload.
That's the bottom line for your tissues.
Which brings us to the question of where these bags come from. Section 2 of our outline covers the factory. And this was a journey I didn't expect.
It's a moving target.
It really is. I assumed bone marrow was the answer, period. Full stop.
It's the answer for you and me right now as adults, but it changes dramatically throughout your life. The source material traces this chronological breakdown really well.
It starts in the embryo, in the yolk sac.
Right. In the very early embryo, the very first red blood cells are produced in the yolk sac. And it's important to note, these are primitive cells. They are nucleated. They haven't learned the trick of spitting out their nucleus yet to make more room.
Okay, so that's phase 1. Then what happens?
Then, in the middle trimester of gestation, the liver takes over. For a significant part of fetal development, the liver is the main blood factory.
My liver was making my blood.
It was. The spleen and lymph nodes help out a little bit. They contribute some cells, but the liver is doing the heavy lifting.
And then there's another switch.
Another switch. In the last month of gestation, and immediately after birth, the factory moves again. And this time it moves exclusively to the bone marrow.
And it stays there for the rest of our lives.
It stays in the marrow, yes, but the location of the active marrow changes as we age. And this, to me, is one of the most sobering details in the text.
What's that?
The bone marrow gets fatty.
Fatty marrow. That sounds like a culinary term, but I assume it's not good for blood production.
It's not. It effectively means the marrow becomes inactive. It's replaced by fat. The text points out that by age 20, the marrow in the shafts of your long bones, your humerus, the big bone in your arm and your tibia, your shin bone, it stops producing red blood cells almost entirely.
Wait, really? By 20?
By age 20, it retires. Fills with yellow fat. Now, the proximal portions, the ends of those bones, might stay a little active, but the main shafts are done.
So my leg bones aren't making blood anymore.
Not in any significant way.
Yeah.
After age 20, most of your red blood cells are produced in what are called the membranous bones. The vertebrae in your spine, your sternum or breastbone, your ribs, and the egulia, which are your hip bones. That's where the factory consolidates.
And even that declines.
Even that productivity declines with age. The overall cellularity of the marrow decreases.
So we have a factory that is slowly downsizing over our lifetime, shutting down at satellite locations, and consolidating into the core.
Exactly. It's a slow, steady decline in our reserve capacity, which is one reason why older people are more prone to severe anemia when they get sick or have blood loss. Their ability to ramp up production just isn't what it was at age 18.
Okay, let's look at the worker on the factory floor. The source introduces the master cell, the pluripotential hematopoietic stem cell.
The PHSC, or sometimes just called the hematopoietic stem cell.
Pluripotential. It sounds like a guidance counselor describing a gifted kid.
It really is. You can be anything you want, and that's literal. All the cells of the circulating blood red cells, every type of white cell, platelets, they all descend from this one common ancestor.
It's the Adam or Eve of the entire blood system.
That's a good way to think about it.
So how does it decide what to become? Does it just flip a coin?
It's all about chemical signaling. So, first, the PHSC has to reproduce to keep its own numbers up. It maintains a stable population of itself in the marrow. But then, some of its offspring become committed stem cells.
They pick a major.
They declare a major, exactly. They start down a specific path. For example, a sighted stem cell that is destined to produce erythrocytes is called a CFUE.
Colony-forming unit erythrocyte.
Right. And once a stem cell reaches that CFUE stage, it's locked in. It can't change its mind and decide to become a white blood cell. Its fate is sealed.
So what pushes it down that path? What makes it commit?
It's a class of proteins called growth inducers and differentiation inducers. And this is a really neat distinction the text makes.
Growth versus differentiation.
Exactly. First, you have growth inducers. The big one mentioned is interleukin-3. Think of interleukin-3 as the general coffee supply for the office. It promotes growth and reproduction of all the different types of stem cells. It doesn't tell them what to do, it just keeps the whole colony alive and buzzing.
Okay, so it's the keep working signal.
Right. But then you have the differentiation inducers. These are specific proteins that tell a cell, okay, stop just growing randomly and start turning into a red blood cell specifically, or a neutrophil, or a lymphocyte.
And these inducers are controlled by outside factors, like supply and demand.
Precisely. Low oxygen in the body triggers the release of the specific differentiation inducer for red blood cells. A bacterial infection triggers the inducers for the white blood cells that fight bacteria. The body orders exactly what it needs from the stem cell menu.
That is an incredibly elegant system. I want to walk through the assembly line now.
Yeah.
Because the text breaks down the genesis of these cells in section 3. And it's not as simple as stem cell becomes red blood cell. It's a metamorphosis.
It's closer to a caterpillar turning into a butterfly. There are distinct, identifiable stages, and the names they give in actually tell you what is happening inside the cell chemically. It all starts with the proerythroblast.
Pro meaning before, blast meaning a sprout or a germ cell.
Right. This is the first cell of the red blood cell series that we can identify. It's a very large cell. It's the raw material that's been stimulated by erythropoietin, which we'll get to.
Right.
But then as it divides, we see the internal machinery change. The next stage mentioned is the basophil erythroblast.
And basophil refers to the dye it absorbs in the lab, right? It stains with basic dyes.
Yes, it stains blue with basic dyes. But why? That's the cool part. The why is what tells you the story. It stains blue because the cell's cytoplasm is packed with ribosomes.
The little protein factories?
Exactly. The cell is essentially building the factory floor. It hasn't started making the product, the hemoglobin, in any serious quantity yet. It's just installing all the conveyor belts and assembly line machinery. That's why it's blue. All that RNA in the ribosomes stains blue.
Okay, so step 1, build the factory.
Step 2, start production. This is the polychromatophil erythroblast stage. Poly meaning many, chroma meaning color.
Many colors.
Right, because under the microscope, you see a mix of blue from the remaining ribosomes and pinkish red from the hemoglobin that's starting to accumulate.
The product is starting to pile up on the factory floor, mixing with the machinery.
A perfect description. And then finally, the orthochromatic erythroblast stage. The factory is now so full of hemoglobin, remember that 34% limit we talked about, that the machinery is getting in the way. It's almost pure red now.
So what happens to the factory management? The nucleus.
This is where things get really wild. The nucleus, which contains all the DNA, all the blueprints, condenses down to a tiny, hard, dark little knot. The cell essentially decides, I don't need the blueprints anymore. I don't need the factory manager. I just need to be a shipping container.
And this leads to the most, metal moment in cell biology. The extrusion of the nucleus.
It literally spits out its own brain. The cell pushes the condensed nucleus up against the cell membrane and ejects it. It's an incredible process to watch.
And the cell that's left is called a reticulocyte.
A reticulocyte, yes.
Why reticulocyte? What does that mean?
It's because it still has a few scraps of the factory left inside, some mitochondria, some remnants of the Golgi apparatus. This leftover basophilic material forms a little mesh-like network or reticulum that you can see with special stains.
And this is the cell that enters the bloodstream, the one that's not quite mature.
This is it. This is where the process of diapetesis happens. The reticulocyte, which is still a bit of a flexible blob, squeezes through the pores of the capillary membrane in the bone marrow to enter the circulation.
Squeezing through the pores. That's diapetesis.
Correct. It's an active process. It crawls through.
And then it matures in the blood.
Yes. Once it's in the bloodstream, that remaining basophilic material, the reticulum, disappears within 1 to 2 days. The last bits of the factory are broken down and recycled. And then finally, you have a mature erythrocyte ready to work for 120 days.
So if a doctor sees a lot of reticulocytes in my blood work, a high retic count, what does that mean?
It means your bone marrow is working overtime. It's a sign of rapid red blood cell production. The factory is pumping out new cars so fast that it's pushing them onto the highway before the paint is fully dry, so to speak.
Ah, so it's a key diagnostic clue for something like recent blood loss or hemolysis.
Exactly. It tells the physician that the factory itself is responding to a problem. If you're anemic and your red count is high, your marrow is working. If you're anemic and your red count is low, that points to a problem with the factory itself.
I want to skip ahead to the regulation system because this was the part of the reading that actually surprised me the most.
The control mechanism.
Yes, I assumed if we need more blood, the bone marrow, the factory would know. It would sense the inventory is low somehow. Local control.
That would make sense logically, right? The producers should know what the stock levels are.
Right, but the text says the manager isn't the marrow, it's the kidneys. And I honestly stopped reading for a second and thought, why the kidneys? They're the waste filtration plant. Why are they in charge of oxygen logistics for the entire body?
It's one of those beautiful, non-obvious bits of evolutionary engineering. Think about what the kidney actually does. It's not just a passive filter like a coffee filter.
No, it's actively pumping things.
It is actively pumping enormous quantities of sodium and other ions against steep concentration gradients. That process requires a tremendous amount of energy, of ATP.
Which requires oxygen.
Massive amounts of oxygen. The kidney consumes oxygen at a rate way higher than most other tissues relative to its weight. It is perpetually on the edge of hypoxia. It's incredibly sensitive to the gap between oxygen supply and oxygen demand.
So it's the canary in the coal mine for the whole body.
It is the perfect sensor. If total body oxygen levels drop even slightly, the kidney feels it first and it feels it most acutely.
And the mechanism the text describes here is fascinating. It's not a vague sense. It's a specific molecular switch.
It's pure molecular clockwork. It's a transcription factor called HIF1.
Hypoxia-inducible factor 1.
That's the one. In the renal cells, when oxygen levels are normal and high, HIF1 is produced, but it's immediately broken down. It's tagged for destruction as soon as it's made. It's essentially invisible to the cell's machinery.
It's held in check.
It's held in the off position by the presence of oxygen. But the second oxygen levels drop,
The destruction stops?
the destruction stops. The factor is no longer tagged for degradation, so it starts to accumulate in the cell. It travels to the nucleus, it binds to a specific spot on the DNA called the hypoxia response element, and it turns on the gene for erythropoietin.
It's a switch that is naturally held down by oxygen. Remove the oxygen and the switch flips up.
A perfect analogy.
And this produces the hormone erythropoietin, or EPO, the famous EPO.
The very same. And the text is clear. 90% of all the EPO in your body comes from the kidneys. The other 10% or so comes from the liver.
This explains something critical then. This explains why people with chronic kidney failure almost always get severe anemia.
It's the without kidney problem, as the book calls it. If your kidneys are destroyed, or if they are removed for a transplant, you lose the factory manager. You lose 90% of your EPO production.
And the liver can't pick up the slack.
The liver can only produce about a third to maybe half of the needed EPO at best. So these patients become severely anemic. They require injections of synthetic recombinant EPO to survive. It's a lifeline for them.
How fast is this system? If I, say, travel to a high-altitude city, how quickly does this kick in?
The EPO production starts within minutes to hours. It's very rapid. The kidney senses the hypoxia, HIF1 accumulates, and you start pumping out EPO. The hormone level in your blood peaks in about 24 hours.
But I won't feel better in 24 hours.
No, because remember the assembly line we just talked about?
It takes time to build a car.
Right. EPO stimulates the production of pro-erythroblasts from the stem cells, and it speeds up all the subsequent stages of maturation. But even with that acceleration, new red blood cells, new reticulocytes, don't start appearing in the circulating blood in large numbers for about 5 days.
So there's a 5-day lag between the order and the delivery.
There's a lead time on the factory, which makes sense. You can't just materialize millions of complex cells out of thin air.
Okay, so we've got the signal, which is EPO. We've got the factory, the bone marrow. Now we need the raw materials. Section 5 covers quality control and maturation. And aside from iron, which is the obvious one, what else do we need?
You need 2 absolutely critical vitamins. Vitamin B12, also called cyanocobalamin, and folic acid.
And these aren't for making the hemoglobin, right? This isn't for the cargo. This is for the cell itself.
This is for the factory machinery. Specifically, they are essential for the synthesis of DNA. Yes. Both are required in different ways for the formation of thymidine triphosphate, which is one of the 4 fundamental building blocks of DNA. A, T, C, and G. They're essential for the T.
So without B12 or folic acid, you can't make DNA properly.
And if you can't make DNA, you can't duplicate your chromosomes. And if you can't do that, the cell can't undergo mitosis. It can't divide.
And the bone marrow cells are dividing constantly.
They're some of the most rapidly growing and reproducing cells in the entire body. So if you slow down DNA synthesis, they are one of the first cell lines to fail.
What does that failure look like? Does production just stop entirely?
Not exactly. It's a stranger process. The cells still try to grow, they mature, they make hemoglobin, their cytoplasm gets bigger and bigger. They're getting ready to divide, but they can't complete the final step because the DNA isn't ready.
So they just get stuck.
They get stuck in a bloated state. You end up with these very large, flimsy, often oval-shaped cells called macrocytes.
Macro meaning big.
Yes. And this condition is called maturation failure. These macrocytes can carry oxygen, some of them, but they are incredibly fragile. They have a flimsy membrane and a short lifespan, so they break easily in the bloodstream, leading to a severe anemia.
And the text goes into a very specific, almost bizarre story about vitamin B12 absorption.
It's not straightforward.
No. It's not as simple as just eating a steak. It reads like a spy novel where the VIP needs a dedicated bodyguard to get through customs.
It is a fascinating mechanism, and it all starts in the stomach. The parietal cells in the lining of your stomach secrete a glycoprotein called intrinsic factor.
Intrinsic factor, that's the bodyguard.
That's the bodyguard. Vitamin B12 is sensitive. If you just ate it, it could get broken down by digestive enzymes. So intrinsic factor binds tightly to the B12 in the stomach. This complex protects the B12 from digestion.
So it puts it in an armored car.
Exactly. Then, this armored car, the B12 intrinsic factor complex, travels all the way through the stomach and the small intestine to the very end, to a section called the ileum.
And there's a specific door there waiting for it.
A specific docking station. There are special receptors on the brush border of the intestinal cells in the ileum that only recognize the intrinsic factor. The B12 can't get in on its own. It needs the bodyguard's VIP pass.
And then it gets in.
The receptor binds the complex, and the cell membrane engulfs the whole thing through a process called pinocytosis. It's an incredibly specific and secure transport system.
So this implies that if I have a stomach problem, I could end up with a blood problem.
Absolutely. The text specifically mentions atrophic gastric mucosa, a condition where your stomach lining thins out and stops working, often due to chronic inflammation or an autoimmune issue.
No stomach lining, no parietal cells.
No parietal cells, no intrinsic factor, no bodyguard. So you can eat all the B12 you want in your diet, but you won't absorb any of it. It just passes right through you.
And this leads to pernicious anemia.
Pernicious anemia. The word pernicious means harmful or fatal in a gradual, subtle way. And historically, it was fatal. Before we understood this mechanism, people would just waste away from this B12 deficiency, and nobody knew why.
Now let's get to the chemistry set. Section 6, we are making hemoglobin. Factor 33-5 in the text outlines this recipe, and it's quite a chain reaction.
It is a beautiful piece of biochemistry, and it starts with surprisingly simple, common ingredients that are already floating around in the cell.
What are they?
It starts with succinyl-CoA, which is a byproduct of the Krebs cycle, just basic cellular energy metabolism, and glycine, which is one of the simplest amino acids.
So the body takes a piece of metabolic waste and a simple protein block.
And in the mitochondria, it combines them to form a molecule called a pyrrole.
A pyrrole, a little ring structure.
A little ring. Then you take 4 of these pyrrole rings and you combine them to form a much bigger ring structure called protoporphyrin IX.
The names are definitely getting longer.
They are. Now, here is the magic moment, the absolute heart of the process. You take that protoporphyrin IX molecule and you add 1 atom of iron.
That creates heme.
That creates heme. That's the active ingredient. The hem molecule is what actually binds the oxygen. The iron atom at its center is the magnet for the oxygen molecule.
But that's not hemoglobin yet.
Not yet. That's just the prosthetic group. Next, you take a heme molecule and you combine it with a long folded polypeptide chain called a globin. That gives you 1 hemoglobin chain, which can be an alpha chain or a beta chain, depending on the amino acid sequence.
We aren't done yet, are we?
Nope. The final step. You take 2 alpha chains and 2 beta chains, you combine them into this massive 4-part structure, and you get hemoglobin A. That is the primary type of hemoglobin in an adult human. A molecular masterpiece with a weight of 64,458.
That is a massive molecule built from scratch inside a developing cell in the bone marrow.
It is, and it shows you again how fragile the system is to tiny errors. Which brings us to sickle cell anemia.
Yes. The text mentions this as a tiny error with catastrophic consequences.
A microscopic error. It's a single-point mutation in the gene that codes for the beta chains. Just one DNA base is wrong.
And that results in?
That results in the amino acid valine being substituted for glutamic acid at one specific point in that long beta chain. One swap in a chain of hundreds of amino acids.
One wrong Lego brick in a giant castle.
A perfect analogy. But that one wrong brick changes the chemical properties of the entire molecule. When this abnormal hemoglobin is exposed to low oxygen, it becomes unstable. It starts to crystallize.
It turns into solid crystals inside the red blood cell.
Long needle-like crystals or filaments. These crystals are rigid and they physically push against the inside of the cell membrane, stretching it out. The cell goes from being a flexible biconcave disc to a rigid, brittle sickle shape.
And that sickle shape can't squeeze through capillaries.
It can't. It gets stuck. And worse, the sharp crystals often rupture the cell membrane. It creates this vicious cycle of blockage, oxygen deprivation, which causes more sickling, and widespread cell destruction, all because of one amino acid swap.
That is both terrifying and fascinating. Now, you mentioned iron being the magic ingredient for heme. Section 7 calls iron metabolism the bank.
Which is a great way to think about it because iron is a precious and dangerous commodity.
Dangerous.
Very. Free iron in the body is toxic. It catalyzes the formation of free radicals that damage cells. So the body has a very sophisticated logistics and storage system to handle it, to make sure it's always bound to something.
Okay, so let's meet the players in this system. Who is the transport? Who's the armored car?
The armored car is a protein called transferrin. When iron is absorbed from the gut or released from old recycled cells, it immediately binds with a protein in the plasma called apotransferrin to form transferrin. It travels safely through the bloodstream in this bound state.
And the storage, the bank vault.
The vault is ferritin. Inside the cells, especially in the liver and the bone marrow, iron combines with a protein called apoferritin to form ferritin. This is storage iron. It's kept in a soluble, non-toxic form, but it can be accessed quickly if the body needs it.
But the text mentions something else called hemosiderin. What's that?
That's the overflow storage, the mattress stuffing. If the amount of iron in the body is more than the apoferritin storage pool can handle, the excess starts to clump together into large insoluble clusters called hemosiderin.
And that's bad.
It's not ideal. You can actually see these microscopic iron clumps in tissue biopsies. It's a sign that the iron storage system is completely saturated and overwhelmed.
So how do we regulate the total amount in our body? Do we just pee out the excess if we eat too much iron?
Surprisingly, no. The body's actually very bad at getting rid of excess iron. We have very limited ways to excrete it. Men lose about 0.6 milligrams a day just through cells sloughing off the skin or in the gut. Women lose more, on average about 1.3 milligrams due to menstrual blood loss. But we don't have an active dumping mechanism.
So how do we stop from, rusting from the inside out?
We regulate it at the front door. We regulate the intake. We control absorption at the level of the intestinal cells. If the body is saturated with iron, if the transferrin is fully loaded, and the ferritin stores are full, the absorption of iron from the food you eat basically stops.
The gut just blocks it.
It blocks it. It's a feedback mechanism called mucosal block. The body says no vacancy, and the door is shut. It's a very smart, proactive system.
Let's move to Section 8, destruction, the end of the line. These cells work incredibly hard. They get squeezed, battered, and chemically stressed for months. How long do they last?
The average lifespan is about 120 days, 4 months.
And what happens at the end? Do they just dissolve in the blood?
No, they fail a physical fitness test. As they age, their metabolic systems start to break down. They run out of key enzymes, they produce less ATP, their membrane becomes fragile and less pliable than it was when it was young.
And then they hit the final exam.
They hit the spleen.
The spleen.
The spleen is often called the graveyard of red blood cells. Its internal structure is a maze, a meshwork called the red pulp. For blood to get through the spleen, it has to squeeze through tiny gaps in this meshwork, through the trabeculae. And those gaps are only 3 micrometers wide.
Wait, you said earlier the RBC is almost 8 micrometers wide.
Oh, exactly. A young, healthy, flexible bag can deform and squish its 8 micrometer self through that 3 micrometer gap. It's a tough squeeze, but it makes it. But an old, fragile, rigid, 120-day-old cell...
I can't make it.
It ruptures. It bursts right there in the spleen.
So it's a quality control test that literally kills the failures.
Brutal, but incredibly effective. It ensures that only the most functional, flexible cells remain in circulation.
And what happens to the debris? The broken bags?
No waste. We recycle everything. The body is the ultimate recycler. Macrophages big eater cells, specifically the Kupffer cells in the liver, and the macrophages in the spleen ingest the hemoglobin from the ruptured cells.
And they break it down into its component parts.
They do. They're like a cellular chop shop. They expertly strip the iron out of the Ham molecule. That iron goes right back to transfer in the armored car to be taken back to the bone marrow and built into a new generation of cells. We recycle almost every single atom of iron.
That's incredible. And the rest of the molecule? The porphyrin part?
The porphyrin ring structure, the part that's left after the iron is removed, is converted through a series of steps into a pigment called bilirubin.
Bilirubin, I've heard of that. That's what causes jaundice.
Exactly. The liver secretes this bilirubin into the bile, and it's eventually excreted. If your liver isn't working, or if red cells are breaking down too fast, bilirubin builds up in the blood and makes your skin and eyes look yellow.
This brings us perfectly to Section 9, anemia. When the system fails. The text defines anemia simply as a deficiency of hemoglobin or a deficiency of red blood cells. But there are so many ways for the system to break.
So many. We can break them down by the mechanism of failure. Type 1 is the most straightforward.
Yep.
Blood loss anemia.
A hemorrhage, a cut, an ulcer, something like that.
Right. If you have an acute hemorrhage and lose a lot of blood, your body replaces the plasma, the liquid portion, very quickly, within 1 to 3 days. But as we said, the RBCs take weeks to replace, so your blood is diluted for a while.
And what if the loss is chronic? Like a slow, persistent bleed in the gut from an ulcer or a tumor?
Then you have a different problem. You are constantly losing iron. Over time, you can't absorb enough iron from your diet to keep up with the losses. So the bone marrow is starved for this critical ingredient.
And the cells it produces are deficient.
They're small and pale. The body tries its best, but it produces cells that are smaller than normal and don't have a full complement of hemoglobin. This is called microcytic hypochromic anemia.
Microcytic for small cell, hyperchromic for less color.
Exactly.
Okay, that's type 1. Type 2 is aplastic anemia. This sounds worse. This is when the factory itself shuts down.
It is worse. This is bone marrow dysfunction or aplasia. It means the marrow just isn't producing cells. This can be caused by exposure to high-dose radiation, like from gamma rays, or certain toxic chemicals like benzene or insecticides. It can also be from an autoimmune disease like lupus, where your own immune system attacks the stem cells.
So the factory just closes.
It closes down, and the only treatments are blood transfusions to keep the person alive, or a bone marrow transplant to try and restart the factory with new stem cells.
Type 3 is megaloblastic anemia. We touched on this with B12 and folic acid.
Yes, this is the maturation failure we discussed. Loss of B12, folic acid, or intrinsic factor. The factory is trying to work, but the machinery for making DNA is broken, so you get those big, weird, fragile megaloblasts that die prematurely.
And finally, type 4, hemolytic anemia.
This is where the cells are being produced normally. The factory is fine, but they are being destroyed in the bloodstream much faster than their normal 120-day lifespan.
Dude, cars are exploding on the highway.
An excellent way to put it. This includes genetic conditions like sickle cell, which we discussed. It also includes things like hereditary spherocytosis, a condition where the cells are fragile spheres instead of biconcave discs. They can't handle the squeeze in the spleen, and they just pop.
And there's also an immune type mentioned, erythroblastosis fetalis.
Right. That's when an Rh-negative mother develops antibodies against her Rh-positive fetus. The mother's antibodies cross the placenta and attack and rupture the baby's red blood cells, a classic example of hemolytic anemia.
I want to talk about the physics of anemia, the overall effect on the body. The text mentions viscosity and why that matters so much.
This is a critical point that often gets overlooked. The physiology is fascinating. Blood is normally about 3 times as viscous or thick as water. That viscosity creates resistance to flow.
Okay.
In severe anemia, you have fewer cells, so the blood gets much thinner. Its viscosity can drop to just 1.5 times that of water.
It gets watery.
Very thin. Now think about what that does to your circulatory system. First, lower viscosity means much less resistance to blood flow through the vessels.
So the blood flows faster and more easily.
Much more easily. On top of that, the low oxygen in the tissues from the anemia causes peripheral blood vessels to dilate, to open up, to try and get more blood flow.
So you have thin blood flowing through wide open pipes.
Exactly. The result is a massive increase in venous return to the heart and consequently a massive increase in cardiac output. The heart has to pump furiously 2 to 3 times the normal amount of blood per minute just to keep up.
That sounds exhausting for the heart.
It is. The heart is working at near maximum capacity just while you're sitting at rest. Then, if you try to exercise, your muscles demand even more oxygen. The heart tries to pump even harder, but it literally cannot. It's already redlining. This can lead directly to acute cardiac failure.
Wow. So the heart fails not from a blockage, but from sheer overwork caused by thin blood.
Correct.
Now let's flip the script. Let's go to the other extreme. Section 10. Too many cells. Polycythemia.
Polycythemia. Poly meaning many. Cythemia referring to cells in the blood.
And there's a normal, or at least physiological version of this, right?
Yes. Secondary polycythemia. This is when the body produces more red blood cells in response to a specific trigger. It's a compensation.
What kind of trigger?
The most common one is living at high altitude. If you move to Denver or La Paz, the air has less oxygen. Your tissues become chronically hypoxic.
So the kidney EPO thermostat kicks in.
It kicks in and it stays on. The body says, well, if each truck can only carry less oxygen, I'll just put more trucks on the road. People living at 14,000 feet might have a red blood cell count of 6 to 7 million instead of the normal 5 million. It's a physiological adaptation. It's normal for them.
But then there's the pathological version, polycythemia vera.
Also known as erythemia. This is a very different beast. This is a genetic aberration in the hemocytoblastic stem cells in the marrow.
It's a cancerous-like condition.
It's a myeloproliferative disorder. Basically, the factory goes rogue. The cells that produce red blood cells don't respond to normal feedback. They don't stop producing even when the body has plenty of oxygen.
They just keep churning out cells.
They don't stop. The red blood cell count can hit 7 or 8 million. The hematocrit, the packed cell volume, can go from 45% up to 60% or even 70%.
My God. The blood must be like sludge at that point.
It is. The text says the viscosity of the blood increases from the normal 3 times that of water to as high as 10 times that of water.
10 times. That's like trying to pump syrup or molasses through your veins.
It is. And as you can imagine, this causes huge problems. The capillaries become plugged, the blood flow is incredibly sluggish, the heart has to strain immensely to push this thick fluid around.
And this leads to a really weird symptom described in the text. The patient's complexion.
The ruddy complexion with the bluish tint, cyanosis.
Right, why blue? If they have more red blood cells than anyone, shouldn't they be the pinkest, most well-oxygenated looking people on the planet?
You would think so. It's a paradox. But the reason is the sluggish blood flow. The blood is moving so slowly through the skin capillaries that the tissues have a very long time to extract oxygen from it.
So by the time the blood gets to the venous side of the capillary, it's almost completely out of oxygen.
Almost completely. A larger than normal quantity of the hemoglobin in their skin is deoxygenated. And deoxygenated hemoglobin has a dark blue-purple color. So despite having a massive total quantity of blood, a huge portion of it is deoxygenated at any given time, giving their skin that bluish tint.
That is a fascinating, counterintuitive consequence of the physics. You have too much blood, but you look like you're suffocating.
It illustrates perfectly that physiology is all about balance, homeostasis. Too little is fatal. Too much is fatal.
It really changes how you view the concept of optimization in biology. We as humans, we always think we want more. More oxygen, more energy, better performance. But the red blood cell teaches us that biology is a game of constraints and trade-offs.
It is. It's a system that's been optimized to a knife's edge. You can try to cheat it, like with EPO doping in sports. You can boost your hematocrit to 60% and get a temporary performance advantage. But you do so at the risk of crashing the fluid dynamics of your entire vascular system. You risk stroke, heart attack.
Because you're pushing the viscosity into that dangerous polycythemia vera range.
Exactly. We are already operating at the absolute theoretical limit of the materials. We are at the max hemoglobin concentration, the optimal cell shape, the ideal hematocrit for flow.
It's a closed-loop system that has been refined over millions of years. And here's the thought I'm left with after this whole deep dive. We talked about how the system is regulated by oxygenation, not by simply counting the cells.
Right. The kidney is the thermostat, sensing the oxygen level.
It makes me wonder about our modern environment. Most of us live sedentary lives. We live in climate-controlled boxes at sea level. We rarely sprint. We rarely experience true hypoxia unless something is wrong. We have essentially put this incredibly dynamic, reactive system into a state of, dormancy.
That's a great way to put it. We've silenced the signals. The kidney is never really stressed, so the marrow is never truly challenged to ramp up production. We're constantly driving a Formula One car in a 20-mile-per-hour school zone.
And what happens to a machine that never runs at redline? What happens to a muscle that's never challenged? It gets fatty, it gets weak, just like the marrow in our long bones as we age.
That is a very sobering thought. Use it or lose it, quite literally written into our bones. The capacity we don't demand from the system might just fade away.
That's it for this deep dive into the red blood cell. Thanks for listening.